Donc on va commencer cette journée du 25 octobre par une plénière qui sera en grande majorité, pour ne pas dire totalement, animée et assurée par nos collègues et nos amis américains, puisque j'ai le plaisir d'abord de co-présider cette session avec le professeur MONTOYA de Bethesda, qui est en charge du département médicaments National Institute Drug Abuse, que vous connaissez, puisqu'il a également participé au débat de société hier sur la vape. Je le remercie évidemment de me permettre de briller à ses côtés. Je laisserai aussi la parole à Ivan pour présenter les orateurs qui vont suivre. Mais on va commencer, you're the first. On va d'abord commencer par la première communication qui précisément est celle d'Ivan Montoya, suivra celle de notre ami Marco PRAVETONI de Seattle, Sandra COMER, et puis nous aurons à la fin notre ami Gavin BART, qui également est intervenu hier pour ceux qui étaient là sur la question de la vape, qui, comme vous l'avez, pour ceux qui étaient là hier, vu que ça a beaucoup débattu et mobilisé les opinions et puis les confrontations de preuves scientifiques. Voilà, donc cette session plénière qui est la première, évidemment, de la journée, nous l'avons intitulée Addictologie, Actualités et traitement des addictions. Et donc, ça, nous avons une première partie, ensuite on fera une pause, et puis je passerai la main, évidemment, à mes collègues, les professeurs Marco PRAVETONI sur le craving et Ikro Maremani sur PIS, Bordeaux versus PIS. On est bien d'accord. Voilà. Thank you, Ivan, for you.
Thank you, Amine. Thank you so much for the invitation to present here. This is a place that is now very familiar to me. I've been in this conference several times, so it's really wonderful to be here among many, many friends. So I'm really enjoying this opportunity. So I'm Gavin Montoya. I'm the acting director of the Division of Therapeutics and Medical Consequences of the National Institute on Drug Abuse in Bethesda in the United States. And I'm going to be presenting a general overview of the situation of drug use, substance use disorders, and overdoses in the United States. And then I will be presenting the different portfolios of research that we have for therapeutics development at the National Institute on Drug Abuse, NIDA. So I'd like to advance the slides. Oh, here. Okay, so Jean-Pierre, when I spoke with Jean-Pierre in archives, they told me, well, you will be presenting like the appetizers of a meal, so of a dinner. So here is the menu of one of the local restaurants, and I will be presenting the appetizers for the different portfolios that we have for opioids, stimulants, cannabis, nicotine. I will be talking about neuromodulation, a little bit digital therapeutics and behavioral therapies, the development that we have in NIDA and some of the future directions. So this is the situation of the past year substance use disorder, not just use. This is people who meet the DSM-5 criteria for substance use disorders and mental illness in the United States in adults 18 and older. And as you can see, we have about 19 million people who are using substance use disorders or have any mental illness. But then we have a number of people who have only a substance use disorder, and we have about 44 million adults with any substance use disorder. So we have many, many people in the United States who meet criteria for any substance use disorder. This is according to the National Survey of Drug Use and Health that is conducted by the Substance Abuse and Mental Health Services Administration, SAMHSA, of the United States. Then this is the past year illicit drug use. This gives you or give us an idea of the different drugs that are being used in the past year by people 12 and older. And as you can see, marijuana is by far the largest, with 52 million people approximately using marijuana in the past year, and then followed by pain relievers, and that includes all the opioid analgesics. Then, interestingly, is followed now by hallucinogens, and hallucinogens was usually in the 4th, 5th place, and now it's now in the 3rd place in terms of illicit substances. Then tranquilizers and sedatives, which we unfortunately don't pay a lot of attention, but we have about 5 million people who use illicit tranquilizers or sedatives. Then followed by cocaine, approximately $4.8 million. Then any stimulants, and by stimulants that includes the prescription medications that are used as a stimulants. And then there's a separate category for methamphetamine, approximately 2.5 million people. And as you can see at the bottom, the heroin is 1.1 million people. It's still many, but it is now in the last place in terms of the frequency of illicit use. Then the past year substance use disorder, which is the one that is very important for us as clinicians. And as you can see, about 24 million people have a diagnosis of any drug use disorder. Of course, as alcohol is more than that, 29. And then followed by marijuana use disorder and pain relief use disorder, methamphetamine, stimulants, and cocaine and heroin. So this is the situation right now in the United States, so 24 million. So with those numbers, we see, well, it's a big problem of substance use disorders, illicit drug use. But the worst part of the worst scenario is in terms of overdoses. Unfortunately, the number of overdoses in the United States is still increasing. So we see here, in the last year, ending in March of 2023, there were about 110,000 deaths associated with drug overdoses. It's a frightening number, and this is going on. 100,000 started since the beginning of the pandemic, and before that, the numbers were increasing. So as you can see here in this slide, we see that some of the drugs have decreased, like for example, heroin overdose has decreased. The overdoses due to natural and semi-synthetic opioids, methadone overdoses have decreased. However, the problem is the synthetic opioids, and you probably have heard the problem in the United States specifically and mainly with fentanyl. Fentanyl is a very potent opioid agonist that is about 50 to 100 times more potent than morphine, that has a specific pharmacology that binds very strongly to the receptor, and the overdose with fentanyl is very difficult to treat because it produces very severe respiratory depression. So that is still overdoses with fentanyl are increasing, but what is really interesting here is to see the overdoses associated with the stimulants, specifically with cocaine and methamphetamine. So they are increasing 9.6 and 3.3% respectively. So this is the trend in overdose deaths in the United States since 1999. As you can see, until about 2014, 2015 was relatively stable. 2015, then yes, a dramatic increase. I don't know if I can here. And primary on fentanyl overdoses. So this is the picture right now as of 2023, about 56,000 deaths associated with fentanyl. And for the stimulants, about 40,000 people die from deaths associated with a stimulant overdose. So those are huge numbers, and those are numbers of great concern right now in the United States. So for that purpose, there is a lot of interest at NIDA in the U.S. government in general to develop safe and effective medications and safe and effective treatments for substance use disorders. So those are the medications that are approved by the FDA, that most of you are familiar with: methadone, buprenorphine, naloxone, For relapse prevention, naltrexone. For opioid withdrawal, we have lofexidine. For opioid overdose, we have naloxone or Narcan, and more recently, it was approved an intranasal formulation of naltrexone. It's called OPV. It was with support from NIDA. So that's the new medication in the armamentarium of treatments for substance use disorders. But we don't have right now any medications for the treatment of stimulant use disorders like cocaine, methamphetamine, or prescription stimulants. And we don't have any medication approved by the FDA for treatment of cannabis use disorder. So in this regard, there has been a history of development of medications and treatments in general with support from NIDA. And those are the targets that, in general, the medications development have been using in terms of developing and studying new therapeutics for substance use disorders. One is prevention, using medications to prevent the initiation or progression to a more severe form of substance use disorder, the prevention of complications of the substance use disorders, and prevention of overdose. Then for treatment per se, we have different approaches. One is the abstinence initiation, relapse prevention. Then we have the treatment of intoxications, treatment of drug withdrawal, specifically, and perhaps one of the most important is for opioid withdrawal. Then treatment of craving, treatment of comorbidity, and there are other targets. This is not an exhaustive list. Then we have for rehabilitation, the management of the complications, reduction of risk, and psychosocial rehabilitation. So the goal of this therapeutic development program is to reduce mortality, of course, but also reduce morbidity, improve the quality of life, and improve the family and social life, and reduce the crime. So most of you are familiar with the phases in medications development, and we have for medications development, we usually have a preclinical phase where we test the different compounds in animals. There's an early preclinical phase. Then there's a late preclinical when more data has been collected and the compound is ready to be is getting prepared to be ready to be tested in humans. Then when all the preclinical data is completed and we are able to get permission from the FDA to study the medication in humans, then we move to the clinical phase, and that includes usually 3 phases. Phase 1 is the first in human studies. Phase 2 is the double-blind, randomized controlled clinical trials to demonstrate initial efficacy, and then phase 3 is the more confirmatory type of studies to support an indication of the compound. So with that in mind, this is the portfolio that we have right now of medications for opioid use disorders supported by NIDA. As you can see, it's a very, very large portfolio. Millions of dollars are being invested in great part because of support from Congress that is giving a lot of support to NIDA to advance the development of medications. You can see here that we have many medications that are in early preclinical phase. Then we have in late preclinical. Then we have phase 1, phase 2, and we don't have any medications right now in phase 3 clinical trials, but we have a number of them that are called like new formulations, that they follow a different path for approval. The goal of all this program is to advance the medications towards FDA approval. So our main goal is to get approval for those by the FDA. And as you can see here, in this slide, and this is one of the things that Jean-Pierre and Archives asked me to do, was to kind of introduce what you are going to be seeing here during this meeting. So you can see here that there will be presentations during this meeting, and we have here in the podium, we have Sandra COMER and Marco PRAVETONI, who are going to be talking of those that are of some of those that are in yellow here, but specifically one that we are very proud of their work, and we are very proud that for the first time we have an anti-opioid vaccine. Thanks to the work of Marco and and Sandy, we have now an oxycodone vaccine that, as you can see here, is in phase 1 clinical trials for the first, so testing for the first time in humans a heroin, an anti-opioid vaccine. We have other anti-opioid vaccines that are in the pipeline, or heroin fentanyl vaccines. Some of this work is also being conducted by Marco, but we have also some in early phases. So we don't have any in phase 2 or phase 3, and our goal is to advance those vaccines. And in addition to vaccines, also we have monoclonal antibodies. So the goal is to advance them towards FDA approval. We have also here, and I highlighted this compound because I know that Indivior is here, and I spoke with someone from Indivior, that we are also supporting Indivior. Believe it or not, we support many pharmaceutical companies. As you can see, many of those products that are in the pipeline are products that we are working and funding pharmaceutical companies to advance the development of those compounds. So you may ask, well, why the government supporting pharmaceutical companies? The answer is very simple. The answer is that, unfortunately, substance use disorders is not a major priority for pharmaceutical companies. They don't think that the investment is they're going to get the return to the investment that they can get with other diseases in medicine like cancer or infectious diseases. So the investment of industry in substance use disorders is very limited. So for that reason, NIDA has a tradition of supporting industry and supporting research with industry. And that's the way that we, for example, supported the development of buprenorphine. That has been a major contributor to improve the life of many people, and naltrexone and many others, and lofexidine. So that is one, and then we have this group here that is there's going to be a session later, pressing the wrong button. Here, it's going to be a session on long-acting formulations. So we have here multiple long-acting formulations that are being developed of multiple compounds. Most of them are with naltrexone, but we also have others with buprenorphine. There is, for example, this long-acting formulation of oral buprenorphine that we are very excited about, that we can give an oral, a capsule to an individual and be covered for 1 week. I know we have weekly injections and we have monthly injections of buprenorphine, and I know that there will be presentations during this meeting about them, but we think that not only having this weekly oral formulation, because as you know, many injection drug abusers are very afraid of needles, right? So the oral formulation is something that can be really an important contribution, and also an oral long-acting formulation of methadone. I mentioned namifene implant and other medications. Then here is the portfolio of medications that are being supported for stimulant use disorders. It's not as robust as we have for opioids, because most of the investment from Congress through what is called the HEAL initiative. HEAL stands for Helping to End Addiction Long Term. It's a very big initiative that is the one that is supporting most of those studies that I just presented. But the HEAL initiative is also supporting some cocaine, some development of medications to treat stimulant use disorders. And this portfolio is also growing. You can see we have here some in the early preclinical, including some biologics, like a cocaine hydrolase. We have here a late preclinical, and including some anti-methamphetamine monoclonal antibodies. We have in phase 1 a couple of cocaine vaccines, and then we have in phase 2, in terms of biologics, some anti-methamphetamine monoclonal antibodies for methamphetamine overdose and for methamphetamine use disorder. There are also some medications that we call repurposed, like, for example, cariprazine, that is approved for schizophrenia, or mirtazapine, approved as an antidepressant, or clavulanic acid is a antibiotic, or ketamine. So we have multiple that are being tested in phase 1B and also multiple in phase 2 as repurposed medications. Unfortunately, we don't have any in phase 3 clinical trials. So our goal is to push this portfolio towards phase 3 to hopefully have someday, maybe before I retire, after working for 30 years in trying to find a medication to treat cocaine or stimulant use disorders, and we still haven't been able to find any, but it's been a very elusive target. So those are some of the ones that I mentioned in terms of biologics, the ones that are being supported now by NIDA. Then we have here, this is the portfolio of what is medications to treat cannabis use disorder. As I mentioned, it's a big public health problem. Many people with cannabis use disorder in the United States. Unfortunately, the portfolio is not as robust as for stimulants or for opioids, but still, we have some. But the interesting part is that we have a couple of some medications that are already in phase 2 and one in phase 3 clinical trials. And those two are going to be presented during this conference. So it is really nice to see that this is progressing. And the one is this fatty acid hydrolase inhibitor, the Phi inhibitor, with the group from Yale, Cyril D'Souza, and the other is this compound that is also from a company from here, from France, Aelis Pharmaceuticals. We are funding this company through a collaboration with Columbia University. Francis Levin is the PI at Columbia. And right now, this compound is in phase 3 clinical trials. We had another study with varenicline, that medications to treat nicotine dependence. But right now, the results, I saw some preliminary results, and it's also very, very encouraging. So although we don't have that many medications in the pipeline for cannabis use disorders, we have very promising medications as those that I just mentioned. Then this is the pipeline for nicotine, and the same thing. We don't have anything in phase 3. It's a pipeline that, although there are safe and effective and FDA-approved treatments for nicotine use disorders, we still have a big problem with many people trying to quit. And we had yesterday the debate about e-vaping. So there's many people who are worried about their nicotine dependence and want to quit. But one of the projects that I want to highlight that is going to be discussed during this conference is this one with psilocybin, with Matthew Johnson from Johns Hopkins has been funded to do studies to advance psilocybin as a potential treatment for smoking or for nicotine use disorder. Now, this is the portfolio of devices. I didn't highlight the ones that are going to be presented here because there will be several presentations during this conference on neuromodulatory devices. Actually, yesterday I met someone from BrainsWave, that one of the companies that in the United States we are supporting, and we have multiple neuromodulatory devices that are being tested. The pathway for approval of devices with the FDA is different than from medication. So I think there's more hope that there will be more FDA approved devices for treatment of substance use disorders in the near future. And finally, it is not less important than the rest of the portfolios, is the portfolio of behavioral therapy development. Our division has a long tradition of supporting research and advancing the development of behavioral therapies, and we use a process similar to what is used for medications. But instead of calling them phases, we call them stages. So we have the medications, the behavioral therapies are being developed in stages. The stage 1 is when, like, first theoretical or some preliminary data is collected to develop a behavioral therapy. Stage 2 is more the first efficacy, and then the stage 3 is more like the large clinical trials to demonstrate the efficacy. And following this process of stages, my division has been able to support, like, for example, contingency management or motivational interviewing and other therapies that have gone through this process of development and are being right now in the implementation phase, being implemented by different payers or clinics. And right now, as I said, we don't have the approval process from the FDA, but the approval process, the equivalent is like getting disseminated and being adopted by treatment programs. So those have been quite successful. So in terms of, just to finish, future directions, we have a robust portfolio for opioid use disorders and overdose. We want to move more towards NDA, towards getting approved by the FDA. As I mentioned, we recently had intranasal nalmefene approved. We want to move more, or you want to have more candidates for stimulant use disorders and cannabis use disorders. We want to have more nicotine. We are very excited and waiting for the results of the studies with psilocybin. And with devices, transcranial magnetic stimulation is one that is of great interest. The other one of great interest is the low-intensity focused ultrasound. I don't know if you have seen the news, but the results of the preliminary studies with that intervention are very, very promising without needing, so it's using ultrasound to focus on certain areas of the brain to treat substance use disorders. Really, really interesting data. We're very excited about that. And as I said, we continue the support of behavioral therapies, and we think that psychosocial interventions are a critical part of the treatment of substance use disorders and the success of patients with those disorders. Thank you for your attention, and I'll go on. Okay.
Thank you, Ivan. On a des interventions plutôt denses. Je vous propose de garder les questions à la fin. Je me suis entretenu discrètement avec les orateurs. Dans la mesure où c'est la même thématique, le mieux, c'est de garder les questions à la fin. Puis je vais donner tout de suite la parole au professeur Marco PRAVETONI de Seattle, qui va poursuivre un peu dans la même veine sur les avancées en matière de prise en charge. Marco.
Merci beaucoup. Wonderful being here, like in Biarritz. Thank you so much, for inviting us and giving the opportunity to talk about, our efforts in this space. And thank you, Ivan, for setting up the stage for this particular presentation. So today, I'm going to focus on some of our work on the development of vaccines and antibody-based therapies for opioid use disorders and overdose. I do have conflict of interest related to patents and disclosure related to the materials that I'm going to show today. As Dr. Montoya gave an outline about how opioid use disorders and overdose in the United States now reach epidemic proportion, and now that is essentially the result of an ever-changing landscape of drug use and risk patterns that keep evolving, especially in the presence of fentanyl analogs, and mixtures that often see opioids mixed with non-opioids such as methamphetamine, psilocybin, and so forth. As far as current medication goes, these are safe, effective, but they seem not just to be enough to curb the current opioid overdose epidemic in the United States. There is a problem of access to treatment, but also the limited number of approved pharmacotherapies may be an issue. What are we doing in this space? Supported largely by NIDA, as Dr. Montoya was saying. We've been working for a decade or so on development of vaccines and monoclonal antibodies to either treat or prevent opioid use disorders and overdose. Through either active or passive immunization, vaccines and antibodies will provide the patient with anti-opioid antibodies that will selectively bind and neutralize the target opioid of interest, and by binding the target opioid will prevent its crossing through the blood-brain barrier, therefore reducing the amount of drug, so the brain concentration of drug, and therefore reducing drug effects such as reward, possibly relapse, toxic effects such as respiratory depression and bradycardia associated with fatal overdoses. And we have demonstrated some of these effects like in mice, rats, and other larger animal models, and now, as Dr. Comer will explain later, in actual human subjects. We think that like vaccines and monoclonal antibodies against opioid use disorders and overdose will provide a longer-lasting, safe and effective alternative to current medication, not to replace them, but to provide physicians and patients with more options. You would have pharmacotherapies plus biologics, so that will hopefully enable reaching like wider population segments, like people with an opioid use disorder, substance use disorder, potentially at-risk non-users. And due to their selectivity, these approaches do not interfere with current medications for pain and critical care, and so they could really be used like in combination with existing medications. So for instance, an heroin vaccine could be combined with methadone, or an anti-fentanyl monoclonal antibody could be combined with naloxone, Narcan, so to speak. Just to give you like a snippet of what we are doing in this area, this is just a selection of like some of the relevant action of these vaccines, specifically in the space of fentanyl and fentanyl analogs. This is our, just as an overview, this is our pipeline of vaccines for OUD and overdose stage that ranges from early discovery to development, pre-IND stage, manufacturing, GLP toxicology, IND stage, and phase 1. Our most advanced product is an oxycodone vaccine that targets oxycodone, hydrocodone, oxymorphone, for which we obtained an IND in 2020, and we are currently doing a clinical trial that is led by Dr. Comer, and she will talk about that later. We are also moving forward with manufacturing with a heroin and a fentanyl vaccine that target their intended drug and metabolites. For example, for a heroin vaccine, we just started last week the GLP toxicology in Canada. Other leads that are moving forward are the carfentanil vaccine, for which we have complete preclinical proof of concept studies. And then we have like a pipeline of next generation vaccines that are advancing either new technologies or new targets or new strategies to actually target multiple opioids and stimulants at once. This is the platform that we are actually using to make vaccines against opioids, but can be used as well to make vaccines against any other drug or small molecule target. There are individual components that can be formulated together. There is a drug-based aptin. Here is derived from oxycodone and hydrocodone, which through a linker, they are conjugated to a larger immunogenic carrier, which is typically a protein, so that the immune system would actually recognize and mount an immune response against the target. This particular conjugate design can be further enhanced by use of either immunomodulators, such as adjuvants, for instance, or formulated with biopolymers, nanoparticles, and so forth. Or different type of strategies can be used to achieve either monovalent or multivalent vaccination with the goal of inducing like CD4 T-cell dependent B-cell activation to elicit polyclonal antibody responses against the target. The design of the aptin is critical to drive like the affinity and selectivity of the antibody response and train them like to essentially achieve antibodies that will bind the target or like closely related structural analogs, but not off-target drugs. And the aptin design is very important because like that will affect all the manufacturing process, so like the conjugation, scale up, and purification, and so forth. This is just an example of a series like stemming from fentanyl or fentanyl analogs, and some of these compounds may or may not be regulated by the drug enforcement agents. As far as efficacy, this is just an example of how vaccines can prevent pharmacological effects of fentanyl and some of the selected analogs. In vivo, these are rats that have been immunized, actively immunized with a small series of vaccines containing different components. Animals that have been previously vaccinated against fentanyl will show the decreased response to fentanyl in various tests. Here we are showing antinociception, respiratory depression, bradycardia. And if you're looking at the center panel, animals that have received fentanyl and they were controlled are pretty much like down to baseline breathing by oximetry, while instead animals that have received the vaccines don't seem to be affected. We have now extended these studies to mice, rats, and large animal models with various like modalities of challenging them with different doses of fentanyl and analogs. As far as efficacy long-term, we have looked at paradigms such as like self-administration, which are standard for this type of research. And so these are rats that have been trained for months to self-administer fentanyl. And then we start immunizing them against fentanyl. And then over time, they will decrease their intake. So we'll stop self-administering the drug, or at least like reduce it. And when like challenged with like a dose reduction paradigm, they will not try to compensate. So in other words, like if the dose reduction will not trigger compensation like is seen in the control. And then there seem to be lasting effect of the vaccine as like months later when the animals are reintroduced with the drug represented, they don't seem to reacquire that type of behavior. As far as safety mechanisms, we spend a lot of time trying to understand how safe and how these things work. And so, for example, here we are looking at how anti-drug complexes are inactive. So the drug, like your fentanyl bound to the monoclonal, does not activate the mu-opioid receptor, the human mu-opioid receptor, in a cell assay on the left. And the reason seems to be related to the fact that the antibody is bound deeply into the binding pocket, as shown like by crystallography studies on the right panel. And we also have spent like time trying to understand if some of these complexes will trigger immune responses, but so far we have not really seen anything, so there is no activation of those effector functions that are typically seen with viruses or pathogens, such as like Fc gamma or other type of phagocytosis effector functions. As far as selectivities, these vaccines are selected as selective, and they're also identified and selected and screened for their selectivity for target, but also by their property of not binding like off-target opioids or other critical care medicines. So for example, on the top, we have demonstrated that our lead fentanyl vaccine is not binding anesthetics, both injectable and volatile, or even in the lower panel, the fentanyl vaccine doesn't interfere with naloxone reversal of heroin, effects of oxycodone, methadone doesn't seem to interfere, buprenorphine as well. And so we really think that such vaccines could be combined with existing pharmacotherapies. As far as formulation, as I was mentioning earlier, you can really go crazy, like, and really, make very fancy vaccines, but in reality, you really need to focus on what can be driven through the regulatory path and would be approved by the FDA. So here, this is a study that we published recently, where essentially we show that our fentanyl vaccine's efficacy was selectively improved by use of novel toll-like receptor 7 and 8 agonists, and this effect was seen in mice, rats, and pigs, as shown in these panels. The immunogenicity, so the antibody response was increased. The effect on reducing respiratory depression was increased by the TLR7 and 8 adjuvant, as well as the ability of the vaccine on panel D to reduce the amount of drug that gets to the brain. So this particular formulation was selected for manufacturing and going towards clinical trials. And so these studies are ongoing. So we're scaling up. We're using CDMOs in US to actually make the manufacturing campaign. And we are doing GLP tox studies in Canada and other countries. As far as multivalent vaccination strategies, these vaccines could kind of Lego-like, could be combined so that you could target potentially different type of either structurally distinct or related drugs. This is just an example that we published recently about combining a lead fentanyl carfentanil vaccine and show how that combination in a bivalent vaccine would actually protect these rats against fentanyl carfentanil mixtures, and that effect was seen by reducing the amount of drug, both fentanyl and carfentanil, to the brain. And now we are actually moving towards quadrivalent, pentavalent, hexavalent, multivalent vaccination strategies that actually could target opioids, different type of opioids, as well as psychostimulants such as methamphetamines, for instance. In order to support our product development pipeline, we spent a decade or so looking at preclinical and clinical biomarkers of vaccine efficacy. Specifically, we focus on the immunology, looking at before and after vaccination biomarker or pathways that would either identify new targets to make better vaccines or biomarkers that would allow us to identify who would be a good responders to this vaccine, so that we could either identify who are good candidates or stratify patients in clinical trials. Most of the work has been done in mice, and now that we have clinical trials, we're actually moving this type of work in humans, as Dr. Comer will talk in the next presentation. As a parallel effort to develop vaccines, we're also developing a variety of antibody-based strategies, mainly monoclonal and antibody derivatives, to either prevent or reverse drug overdose toxicity. Compared to vaccines, monoclonal antibodies will provide immediate protection. You don't need to wait for months to build up the immune response. And so monoclonal antibodies can be used in both pre-exposure but also post-exposure treatment and could be combined with naloxone and hopefully could provide better survival, especially when fentanyl or fentanyl analogs are present. Our pipeline of antibodies, similar to vaccines, spans from early discovery all the way towards IND-enabling studies. We have a lead humanized monoclonal against fentanyl and fentanyl analogs. We've done all the preclinical studies in mice, rats, pigs, and non-human primates, and we have started the manufacturing process, and we are planning the GLP tox and clinical trials in the future. Again, this is work that I'm doing all the preclinical manufacturing. Dr. Comer will do the clinical work. And also we have other antibody targets in the pipeline, so antibodies against heroin, oxycodone, carfentanil, and so forth, as well as we are developing a pipeline on next generation formulation, employing either new technologies or targeting new different type of non-opioid drugs. As far as development of antibodies, like in any other field, especially here, colleagues from the HIV vaccine probably they would actually know how this works, obviously, and so we have parallel workflow for high-throughput screening of monoclonal, starting from either mice, humanized mice, humans, and other species. That goes from discovery to development. We start from an antigen-based magnetic enrichment of opioid-specific B-cell lymphocytes from both animal and human samples, and then from there we either do standard hybridoma or more modern sorting paired with PCR sequencing. But the goal is always to isolate essentially antibody binding regions that then can be expressed and scale up and so forth. Antibody engineering strategies will allow us to optimize the PK/PD profile and improve efficacy against the target. And so, for example, here you have an antibody. We are working sort of both sides of the antibody. On the Fab portion, we are actually processing through humanization. We are doing biophysical characterization and trying to understand what biophysical properties will dictate in vivo efficacy. And also, because of X-ray crystallography and structure, we can actually do targeted mutagenesis to further improve binding against the target, as well as the FC portion can be engineered for extending the half-life of these antibodies. This is just an example in rats. These are animals that first are exposed to fentanyl until you will see a behavioral or pharmacological effect. And then the endpoint is reversed by administering either monoclonal antibodies or naloxone. So in the top, you could see, for instance, that rats, after infusion of fentanyl, they go down to roughly 70% oxygen saturation, which in rats is remarkable. And then we reverse with either monoclonal or naloxone. Or the bottle panel is showing how the monoclonal antibody reversal is somewhat in between naloxone IV and naloxone sub-Q. And so these are some of the efforts that we are advancing towards clinical trials. These are just rats. Moving on to larger animals, we develop models in mini pigs where actually you can have fentanyl apnea. So this model uses induction of apnea. So we infuse fentanyl until the animal stops breathing for 2 minutes, and then we reverse with monoclonal or naloxone. On the left, you could see how the respiratory depression is reversed and is comparable to naloxone. And on the right, you could see the pharmacokinetic mechanism. You could see that as soon as monoclonal is applied, there is that kind of almost vertical shoot up of bound fentanyl, and then seem to be underlying the effect that we are seeing on the efficacy side. As well as in non-human primates, we are seeing similar effects where on the left, we have as a control naloxone and seeing how naloxone reverses fentanyl-induced respiratory depression. These are rhesus macaque equipped with an helmet in order to do ventilation studies. And then on the right, we are seeing superimposed on naloxone how the monoclonal antibody has similar effects. And then obviously this is all dose dependent upon what you're using, when you're using the velocity or the speed of infusion, and so forth. Results obviously may change. As I was mentioning before, now that we have done panels of structure-based design, so we have crystallography of a lot of these antibody targets. Using the sequence, the structure, we can actually use computational method or more artificial intelligence, machine learning to actually predict or modify or selectively mutate sites of the antibodies to make them better. That could support either engineering to make more stable, more selective, more effective antibodies, or can inform design of new antibody modalities such as nanobodies or antibody construct that could be potentially delivered either intramuscularly or intranasally. With that, thank you. There is like my team in both Seattle, University of Washington, where I spent like the last 20 years in Minnesota. Also, we have our team there. And then thanks to NIDA, that support all our research. It's been very generous. And then we have our consortium for medication development with Dr. Comer leading all the clinical work. And then we have numerous collaborators across the country and abroad. So thank you very much for your time.
Thank you, Marco. Sandra? Yes. Donc, on va écouter maintenant Sandra COMER, que vous connaissez, puisque c'est une habituée du colloque ATHS, donc, qui est une chercheuse à New York, qui va dans la même lignée, donc nous parler probablement même sur la vaccination et puis sur les...
I'd like to start out by thanking Professor Dalouet and the organizing committee for inviting me here to give this presentation. I'm very excited to hear my other co-presenters also. There's a lot of work that has been done and still needs to be done. So I have no conflicts of interest to report related to the contents of this presentation. I'm just going to jump right in because Marco has, set the background of the work that we're doing. I just wanted to show you some additional data. Today I'll be talking about our oxycodone vaccine, which we have in clinical testing right now. As Yvonne mentioned during his presentation, normally the FDA requires 2 phases of a phase 1 study. So phase 1A is typically done in normal healthy volunteers. And then phase 1B is done in substance users. In this case, we planned to do that, but the FDA came back and said no, they wanted us to start testing in drug users. So all of the clinical data that I'll be showing you will be in that population. These are data that were collected in rats. And I'm just showing you that the oxycodone vaccine produces a dose-related reduction in brain levels of oxycodone. And correspondingly, it produces a dose-related increase in plasma levels of oxycodone. And as Marco mentioned, the drug antibody complex is not pharmacologically active. So this just demonstrates that the antibodies that are produced by the vaccine are preventing the drug from getting into the brain and producing their pharmacological effects. This is also reflected in the analgesic effects of oxycodone in rats. So with increasing doses of the oxycodone vaccine, we see a dose-related reduction in analgesic response. So that was all incredibly encouraging and was used to support the IND, investigational new drug application for the clinical study. This also was used to support our clinical research. So this is also data collected in rats. How do I go back? Yeah. Rats that were trained to self-administer oxycodone. So there's an inverted U-shaped dose-response curve, which is a typical pattern of responding as you increase the dose of oxycodone that's available. Animals will self-administer a maximal dose, and then eventually they start self-administering less, mostly because they are overly intoxicated. But what is important here is that the vaccine suppresses this self-administration behavior. And we know from other studies of medications that if you can suppress rat self-administration, you usually see a good clinical benefit as well. So the preclinical studies showed that the opioid vaccines that we're testing are well tolerated and they produce antibodies to the target opioid. As I showed and Marco showed in his presentation as well, the brain levels of opioids are reduced in vaccinated animals. And correspondingly, the pharmacodynamic effects are reduced in vaccinated animals. So, these were all incredibly encouraging results that we obtained in the preclinical studies. But we have learned a lot from other vaccines that have been developed for treating substance use disorders. So for a cocaine and a nicotine vaccine, they also showed very promising preclinical studies and even early clinical studies, but both of those types of approaches ultimately failed in clinical testing, so the phase 3 studies were negative. And that was mainly due to inter-individual variability in the ability of the vaccine to induce an immune response. So some people had high antibody levels and other people did not. So as a group mean, there was not a significant effect from placebo. So an important part of the work that we're doing now is to try to identify biomarkers that might predict people who would show a good immune response. So I'll talk about those later in my presentation. So this study was funded by NIDA, and it's a phase 1B, 1A, 1B study. So the primary aim of phase 1 studies is safety. So we need to make sure that it's safe and well tolerated in people. In this study, we also examined immune responses. So we measured antibody levels. And we're collecting preliminary efficacy data. So I'll show you those data as well. When we started this study, we had proposed a larger sample size, but the FDA asked us to conduct a pilot study first, and that's what I'll show you today. So the study design is somewhat complicated, but not too bad, so I'll walk you through. There's a screening phase of about a month where we make sure that everybody meets all of the inclusion and exclusion criteria. We give the vaccine dose 4 times during the study at week 0, 3, 6, and 18. The participants live in the hospital for 2-week periods of time, and then we conduct testing. So we test the effects of oxycodone, a different range of doses, and then we use heroin as a positive control. So they have 3 inpatient periods before the first vaccination, after the 3rd one, and then after the booster vaccine dose. Originally, we had proposed an inpatient period for this entire duration, but during COVID, people were unwilling to be in the hospital, so we changed the design somewhat. So we have completed 12 people so far in this pilot study. One is a low-dose cohort, and the other is a high-dose cohort. So half of the participants received active vaccine and half received placebo in each cohort. So these are just the demographics. You can see that they're mostly men. They come from a variety of ethnic backgrounds, and they're usually like between 40 and 50 years of age. So to describe the first aim of the study, these are adverse events that we collected. There were all of the participants in the low-dose cohort experienced at least 1 adverse event. There were 2 serious adverse events in this cohort. One participant experienced multiple non-fatal overdoses during an extended follow-up period at the end of the study. They did not require hospitalization, but they needed naloxone. And another participant reported an abscess secondary to intravenous drug use that was treated with IV antibiotics, and neither was deemed to be related to the vaccine. There were 2 serious adverse events also in the high-dose cohort. One had a planned cholecystectomy, and the other experienced rhabdomyolysis after an intense workout. These also were deemed to be not related to the medication. There was only 1 treatment emergent adverse event. One participant reported soreness at the injection site. So so far in these first participants, the vaccine has been well tolerated. This is the second aim of our study, to look at antibody response. And as you can see, about half of the participants, so 5 of the 12, showed increases in oxycodone-specific IgG titers. So the responders had a mean of 3,900 titers, and the non-responders was much lower at 155. So again, this was also encouraging. So about half of the subjects received active vaccine. We haven't broken the blind yet, so I can't show you the unblinded data, so I'll just show you some data from participants who showed these high levels of antibodies. So what I'm showing you here is the time course of the effect of the challenge drug on pupil diameter. So here is the person received the placebo oxycodone. This is before vaccination. And then when this person received 50 milligrams of oxycodone, you can see that there's a time-related reduction in pupil diameters, which is exactly what you would expect with a strong mu agonist. After the first week after oxycodone administration, there was no change in the meiosis that was observed. But after the third dose, the meiotic effects of this dose of oxycodone were completely reversed. This is really incredible because we almost never see that. And we were really happy to observe this effect. But we need to collect more data to confirm the response. These are subjective responses. Ratings on the left are showing you drug liking. This is a bipolar scale, so 100 means extremely, and like a lot, and this is dislike a lot, so 50 is neutral. All 3 of these participants reported increases in drug liking, and then there was a reduction after vaccination. So this is after the 3rd vaccine dose. A similar pattern of effect was observed with visual analog scale ratings of good effect. So they all reported liking. This is a unipolar scale, so this means not at all, and this is extremely. So there was this enormous reduction in 2 of the 3 subjects, but all 3 showed a reduction in effect. And then just to put this into context, we have other data that have shown that even a 10 millimeter or 10-point reduction in subjective ratings of good effect or high translate into good clinical benefit. So even though with this one participant, there was a 10 millimeter reduction, it looks small, it actually is a clinically meaningful change. And these obviously are very large reductions in effects. So far, we're very encouraged by these results. We presented the data to our Data Safety Monitoring Board, and they recommended progression to the larger main trial, so that's what we're running right now. We also presented the data to NIDA, the team there, and they also approved progression to the next phase. So now I want to switch gears a little bit and talk to you about our biomarker work, because as I mentioned, it's really important to do this work in order to, we're thinking in the future when we get to phase 3 testing. So we're looking at a number of different potential biomarkers that we can try to identify before vaccination that will predict a good immune response afterward. And basically this is what we're looking for. So if we can identify a biomarker that can be correlated with post-vaccination efficacy, then that would be a good place to be. And we would like to enroll, like a subset of these patients who we think will have a good immune response. And this has been demonstrated in Marco's lab in mice. So what he showed was that pre-vaccination IgM B cell activity was correlated with post-vaccination antibody titers. So this is a strong correlation. And the effect was correspondingly related to decreased brain levels of oxycodone. So this is what we're hoping to demonstrate in people. These are the clinical data that we have collected from the pilot study that I was just showing you. So what we found was that the post-vaccination oxycodone-specific B cells was correlated with post-vaccination oxycodone-specific IgG titers. The oxy-specific plasma cells were likewise correlated. So we're currently testing whether pre-vaccination oxycodone-specific B cells in these participants are also correlated in this manner. So now I'll show you some data from a study that we conducted in a separate groups of people. One was in individuals that had opioid use disorder compared to opioid-naive individuals, and none of these participants were vaccinated. So first point in this study is that there's no evidence of increased opioid-specific IgG or IgM levels in people with opioid use disorder versus normal healthy volunteers. So basically that says that people who are using heroin, fentanyl, these other small molecules, those substances are not enough to trigger an immune response. However, you can still see that within each group there's some variability in response, and that becomes important later in the talk. What we did find was that oxycodone-specific naive and switched memory B cells are higher in people with opioid use disorder compared to those who were not. And the ribbon plots on the right show that opioid use increases antibody variable region rearrangements, which is reflected in the fact that we can isolate a moderate to high affinity human monoclonal antibody against opioids. So this is all the work that has been done in Marco's lab, so we're really excited about that. We also found in this study that oxycodone-specific naive, switched and unswitched memory B cells are correlated with both oxycodone and morphine-specific IgM titers. We looked at cytokine levels as well, and we found that they are suppressed in people with opioid use disorder. This has been reported by other people in the field, so this was not a surprising finding that we had. But what we did find was that the frequency of oxycodone-specific B cells correlates with cytokine expression. We also found that heroin use is correlated with cytokine levels. So there's a positive correlation between heroin use in bags per day and cytokine levels. So this is in the blue panels. So there's a positive correlation here. People who use heroin more have generally higher levels of these different cytokines. There's a negative correlation between the years of heroin use and cytokine levels. At first, we were kind of confused about why that might happen, but then we realized that that's normal. As people age, their immune system becomes less functional. So these are just the same cytokine, IL-9 here, showing this positive correlation between the amount of heroin used per day and the years of use as a negative correlation. The same was true with EGF. So I know I threw a lot of data at you, so I just thought it would be helpful to summarize all of this. The mouse studies showed that pre-vaccination B cell activity is correlated with post-vaccination opioid antibody titers. Our human data show that pre-vaccination B cell activity is correlated with cytokine expression, and that cytokine expression is related to opioid use patterns. So we're thinking that maybe any of these, B cell activity, cytokine expression, or opioid use patterns, possibly could be related to post-vaccination opioid antibody titers. So you ask, why is this important? So the clinical implications are that those with high, pre-vaccination B-cell activity or cytokine expression or opioid use patterns, if they produce high titers, then we're hypothesizing that they'll be related to good clinical response. And this is that same figure I showed you earlier. So this will be an enriched population, hopefully leading to a positive clinical trial. But we have a lot of work to do. So I'd like to thank everyone in my lab for all the hard work that they've done, and I'd like to thank Marco for his collaboration. Thank you.
Thank you, Sandra. On va terminer avec, vous avez bien compris, on tourne autour de quelque chose qui est extrêmement important, c'est toute la crise des opiacés qu'on essaye de solutionner par des nouvelles, des innovations thérapeutiques. J'espère qu'on aura le temps, on le prendra pour un peu challenger, poser les questions à nos collègues. On va finir avec Gavin BART qui très clairement pose la question, comment le fentanyl remet en question le paradigme des traitements USA. Thank you, Gavin.
Thank you, and good morning, and thank you, Jean-Pierre, for fitting me into the schedule at this conference. If Yvonne was the appetizer and Marco and Sandy were the entree, then that may make me dessert, although I prefer to be the digestif. I am hoping to contextualize a little bit of what they have said with the situation with fentanyl in the United States, perhaps as a cautionary tale for Europe. Well, I hope this does not happen in Europe. I know it has been happening for a long time in Estonia, but I do worry about the spread globally. I do have funding from NIDA and NIDDK, and no other conflict. So, again, I want to talk a little bit about the basics of fentanyl in the United States, the challenges that the treatment system has been faced with because of this, and some of our evolving approaches, and why it is so important to have new treatment approaches. As Yvonne mentioned, fentanyl is now accounting for the majority of overdose deaths in the United States, about 75,000 last year. Heroin overdoses are, if anything, decreasing and being completely overshadowed by illicit fentanyl. So why fentanyl? I think in this era of global warming and climate change, we may all understand that being a farmer is very challenging, especially if you're a farmer in a war zone, growing an illegal crop, prone to aerial surveillance, and having to transit a bulky and smelly substance for refinement and then transit around the world. From a business standpoint, illicitly manufactured fentanyl, it makes great sense. You are not reliant on climate. You can hide indoors or underground and avoid aerial surveillance. You have a highly potent substance that takes up very little space and is easy to smuggle. And most of the precursor chemicals are legal. The manufacturing is easy. It's just good business sense. And so it has taken over in the United States, and again, perhaps it will take over in the rest of the world. It is telling that in Afghanistan, they've been switching from opium production to methamphetamine production, again, relying on chemistry rather than agriculture. The main dangers of these illicitly manufactured fentanyl, certainly overdose with 75,000 last year, due primarily to an inconsistent drug supply. People who are buying fentanyl don't know if it's 5% fentanyl and 95% another substance, or if it's 95% fentanyl and 5% another substance, so it's hard to gauge tolerance. There are multiple analogs that are on the market that are being manufactured with varying levels of potency. So what a person buys from one day to the next may change in terms of potency. And there are additional adulterants, xylazine, illicitly manufactured novel benzodiazepine substances, and other types of opioids. The overdose is also more severe in terms of the potential for laryngeal spasm, as well as this wooden chest syndrome that makes it very hard to expand the lungs. Fentanyl is so fast-acting and short-acting that it is highly reinforcing and is leading to more frequent use than a traditional opioid like heroin. Further, we're beginning to see that anywhere from 5 to 20% of the cocaine and methamphetamine supply is contaminated with fentanyl, and so there are people who have no intention of using an opioid who are nevertheless being exposed and are at risk of overdose. I suppose the good news may be that we are seeing some shifting from injecting to smoking in some parts of the country, but smoking in and of itself is not reducing the potential for overdose. So who's using fentanyl and why? This is certainly outdated information because increasingly it's hard to find something that isn't fentanyl. It's getting harder and harder to find heroin. But when both were available, the people who were selecting fentanyl, or at least combinations of fentanyl and heroin, tended to be younger, white, to have more comorbid mental illness, and to be greater risk takers, to be injectors and daily injectors at that, and to have insecure housing. This is important as we begin to see the population using fentanyl having greater comorbidities than our traditional population using heroin. Fentanyl and HIV risk. People who are using fentanyl are more likely to engage in the sharing of syringes and other injection equipment. Again, there are more injections per day, especially when combined with other substances, and more overdoses when combined with other substances. The combination with other substances is also producing greater instability, as the graph from work produced by a colleague at Yale and my colleagues at Hennepin demonstrated that people who are using combinations of fentanyl and methamphetamine are more likely to have housing instability than those who use either drug alone. They're more likely to require government financial assistance and to be involved in the criminal justice system than those who use either substance alone. So there's much more social instability than we are traditionally used to in the era of fentanyl when compared to the era of heroin. So we've got a less stable population that has more comorbidities, particularly skin and soft tissue infections, psychiatric comorbidities, non-fatal overdoses, because of the social disenfranchisement, criminal justice involvement, housing instability. These are individuals who are less likely to choose traditional ambulatory care. They aren't necessarily going to treatment centers or doctor's offices seeking help, but they're being seen in emergency situations, in the setting of overdose, in the setting of endocarditis and other infections, or in the criminal justice system, which means we need to shift our approach to when we are initiating and managing treatment. So I'll talk a little bit about some of the approaches. Certainly in the pre-hospital setting, increasingly areas in the United States, the ambulance are carrying buprenorphine on them. So when there is a community response to a non-fatal overdose where naloxone has been administered, the ambulance can then directly administer buprenorphine out in the community in the post-naloxone, post-overdose setting. When that is done, it is more likely to result in ongoing care. Again, these are people who are not necessarily showing up to a doctor's office to initiate treatment, but if it is initiated in the field, it increases their likelihood that they will access care and that they will have a subsequent overdose in the following week. More commonly, emergency departments are initiating buprenorphine and initiating it directly at doses of 18 to 16 milligrams when there is observant withdrawal symptoms, and then discharging people with up to a week's supply of buprenorphine while making connections for ongoing care in the community. And doing this is resulting in a greater likelihood of engagement in care rather than simply providing relief of withdrawal with traditional medications and a referral into the community. This approach of providing larger doses of buprenorphine in the setting of the emergency department does not seem to be resulting in precipitated withdrawal, even when using long-acting injectable buprenorphine within the emergency department setting. As an internal medicine physician, I'm engaged much more in the hospital-initiated care. And again, we are seeing an increased number of hospitalizations related to skin and soft tissue infections. There's been a doubling of opioid-related hospitalizations in the United States, which creates a great opportunity to initiate treatment for opioid use disorder within the hospital by providing any of the available medications, most commonly methadone or buprenorphine, while in the hospital and making arrangements for continued care post-discharge. And certainly doing this, as opposed to simply providing a recommendation and referral to treatment upon discharge, is more likely to engage patients in treatment following their hospital discharge. There are some novel approaches of supplementing buprenorphine or methadone in hospital with short-acting opioids to relieve any co-occurring withdrawal. That's not being managed by the doses of buprenorphine or methadone. And in some jurisdictions, even providing syringe service programming directly within the hospital and providing safe consumption spaces within the hospital for hospitalized patients. Because not doing so will increase the chance of early discharge from the hospital, incomplete antibiotic courses, and more severe presentation upon readmission. There's also been integration of buprenorphine into low-threshold programs, such as syringe service programs. So by providing buprenorphine directly through syringe service programming, it has increased the uptake of treatment as well as the decrease of opioids, and not requiring patients to show up to a treatment clinic in order to initiate treatment. Because of the housing instability, we're also seeing street medicine providers, doctors with backpacks and stethoscopes and other basic medical equipment, prescribing buprenorphine within homeless encampments and to people who are living on the streets. Again, not requiring them to come to a clinic for treatment. And by doing so, we're seeing at least some retention in a very hard to reach and hard to treat population by bringing the treatment to them. These are some of the approaches that we need to explore more, but the challenges that we're facing is because of the very short action of fentanyl and the potency of fentanyl, people are having a very hard time stopping for any significant period of time, and the initiation of buprenorphine has become quite challenging. Certainly, if people are in withdrawal, we can provide large doses of buprenorphine, anywhere from 16 to 32 milligrams on the first day of treatment. But if they are not in withdrawal, often we have to wait, which for the patient can become extremely challenging, and they may not be able to stop long enough to experience withdrawal to try traditional dosing. So we're increasingly seeing modifications of the Bernice method by having shortened low-dose initiations anywhere from 3 days long to 1 week long, where patients are not required to stop their fentanyl use and slowly titrate it up on their buprenorphine, starting at about 0.5 milligram sublingually daily, and then increasing it gradually to when they can stop the fentanyl altogether. But what we are also seeing is that there is such fear of the potential of precipitated withdrawal that more and more people are avoiding buprenorphine, and we are getting increased requests for methadone specifically, which in the United States, because of the methadone clinic system, which is a very dehumanizing approach to treatment, it's very telling when people are asking for something so dehumanizing compared to a medication like buprenorphine, which is much more freely prescribed. Now, methadone in the setting of fentanyl can be started very easily. There is no need to wait for withdrawal. There's no risk of precipitated withdrawal, but the challenge is getting to a therapeutic dose is very hard if we follow the traditional methods of a 30 milligram dose on the first day and increasing 5 to 10 milligrams every 3 to 5 days thereafter. And that places people at risk for ongoing fentanyl use and the potential for withdrawal. So the question is fentanyl withdrawal different? Are people experiencing something that is, one, leading them to request methadone specifically, and two, continuing to be so unstable? We are seeing patients at doses 100 or 120 milligrams of fentanyl complaining of ongoing withdrawal and ongoing fentanyl use, something that we would not normally see with heroin. And work by Kelly Dunn and others have shown that people who have experienced both heroin and fentanyl withdrawal will describe fentanyl withdrawal as a faster onset and being more severe and longer lasting than heroin withdrawal. And in a laboratory setting at Johns Hopkins, again, work by Kelly Dunn, where they have a standardized approach in a human laboratory setting to giving morphine subcutaneously, 30 milligrams, 4 times a day, which traditionally has been very effective at managing heroin withdrawal. In the era of fentanyl, they've noticed some changes. This is data that she presented at CPDD last year and is working on a manuscript for, that despite this protocol, which has been quite successful for heroin, in people who are using fentanyl, they have more severe ratings of withdrawal despite the morphine, both subjective and objective, more elevations in systolic blood pressure, diastolic blood pressure, and heart rate than those treated historical controls with heroin, receiving the same amount of morphine. And when the symptoms are observed and self-reported, there's certainly much more reported anxiety than traditionally is the case with heroin. So there may be something different about the withdrawal of fentanyl that is contributing to the challenges we're having with treating it with buprenorphine and even our traditional doses of methadone. Now, we can try more rapid inductions of methadone. Certainly, Avram Goldstein in 1970 did blinded dosing starting at 30 milligrams and increasing 10 milligrams a day to get to 100 milligrams within a week. He adapted that down to 100 milligrams by day 15, but we're seeing colleagues in the United States now trying approaches of initial doses of 40 milligrams going to 60 milligrams on day 2 and 80 milligrams on day 3, and increasing to 140 milligrams within the next week or 10 days as needed. And that this approach is showing at least more rapid control of symptoms and unfortunately some fairly high relapse rates to fentanyl despite the very high doses and rapid increase in methadone. Which leads to a hypothetical question, is if we look at some of the pharmacokinetic and dynamics of fentanyl, we see that fentanyl doesn't have particularly higher affinity at the mu-opioid receptor than, say, morphine, and certainly has lower affinity than hydromorphone. And the original narcotic blockade studies by Dole using methadone found that doses of methadone could block hydromorphone quite successfully. So why is it that we're seeing ongoing use of fentanyl despite relatively high doses of methadone? One could potentially speculate that perhaps given the differences in withdrawal symptoms, that there may be non-opioid related factors that need to be studied. Fentanyl, unlike morphine, also has affinity relatively equivalent to norepinephrine at the alpha-1 adrenergic receptor. And so I think we have a lot of work to try to better understand some of the instability that we're seeing with fentanyl users despite ongoing doses of buprenorphine and relatively high doses of methadone. So the take-home message is fentanyl has resulted in increased overdoses and acuity within the United States. Hopefully it will not happen here in Europe. We need more research to better understand why fentanyl use is persistent despite methadone and buprenorphine. We need to better understand how to more rapidly titrate methadone to safe doses and effective doses quickly to reduce the risk of overdose in fentanyl users. And certainly, we need research on new models of medication access and approaches, like we heard Yvonne, Marco, and Sandy present today. So thank you very much.
Thank you, Gavin. Bon, il nous a semblé utile de laisser nos collègues qui ont, évidemment, qui ont traversé l'Atlantique pour nous montrer un peu leurs travaux, les perspectives. On va un peu grappiller, si évidemment on m'autorise 5 minutes pour prendre quelques questions. J'imagine qu'elles sont nombreuses. Et si évidemment vous êtes frustrés, ce qui est 10, bon, bah, 10 minutes. Donc si jamais pendant les 10 minutes vous n'êtes pas suffisamment assouvi, vous pouvez évidemment, ils sont encore là, pendant la pause café, leur poser des questions. Voilà. Ils sont à vous. Qui souhaite prendre la parole? Alors, je vous laisse viser les mains levées avec les micros. Et puis, présentez-vous des questions courtes. Les réponses le seront aussi.
Yes, hello, Luis Montaner from the Wistar Institute. Very nice presentations on the vaccine. Short question, is the drug use challenge boost antibody levels? And second, when you showed the third dose, did the titers decay by the time you gave the fourth dose? Meaning, what is the longevity of memory and what is the relationship on drug use to boost the plasma cells to make more antibody?
Yes.
Well, that's a very good question. So, like, we don't really see a boost of the drug. So the drug boosting like antibodies and neither opiate-specific B cells. So differently than infectious or agent and or a protein antigen. And so in people, we don't seem to see that as well. And so yes, there is like a decay. There is a decay like between like the third and fourth, and then like after the fourth vaccination there is a boost. So we see boost effect on opiate-specific B cells when you give the vaccine, but not the drug.
Oui, je ne sais pas, allez-y. You can-
Hello. Thank you for the presentation. I have two questions. Well, actually three. In the new drug development at NIDA, have you been looking into psychedelics like MDMA, ketamine, and I saw you had psilocybin for nicotine for mental health disorders? I've been reading about that there's been some experiments using these types of drugs to help people with mental health illnesses. And with the vaccines, have you considered contacting Department of Corrections? And a lot of people coming out of jails and prisons have high rates of overdose. It would be really great as part of their re-entry to get a vaccine or something like that. And for Dr. Bart, my harm reduction program in New York, we initiated buprenorphine among people presenting at the needle syringe program in withdrawal. So we gave them an option to continue injecting, or they could get a free buprenorphine just for today to try it out, to give them a break from injecting. So we could talk more about that. But thank you.
So I answered the first question is yes, we are, NIDA is open to accepting grant applications and research looking at psychedelics. And I mentioned we are funding psilocybin for nicotine, but there's also studies with ketamine for cocaine. So yes, the answer is yes.
So to answer the second question about the prison population, I think that's a really great suggestion. I think either the monoclonal antibodies, the MABs, or the vaccine could be effective. I think just ethically, there's a little bit complicated about, giving these types of medications to people in prison. That's the only thing we have to kind of work out. But yeah, it's a great idea.
Thank you. Qui c'est qui a le micro? Oui, vous pouvez donner le micro.
Thank you. So I'm a researcher in neuroscience and psychology, so I will have a psychological question, especially for the vaccine. So basically you're training the blood-brain barrier to catch opioids. But my question is, I'm guessing there are some dose-dependent limitations in the sense that when you increase the dose, you overcome the immune response. So what will happen in people with sensation-seeking, impulsivity, anxiety? Won't the vaccine just incentivize overdoses?
So that's a really great question. And I was involved in the development of a sustained release formulation of naltrexone, and that was the same kind of concern that we had there, is that why wouldn't someone just take more of an opioid to overcome the blockade? And what we observed clinically, though, was that people didn't do that. They're subject to the same kinds of economic principles as anyone else. And so if you block the effects of heroin or fentanyl, people don't want to spend more money to get high. So that didn't really happen. Theoretically, yes, it could occur, but we just didn't see it clinically with naltrexone.
Merci.
Right, thank you.
Encore deux questions. Oui, allez-y.
Bonjour, merci. Docteur Journe, addictologue à Paris. Une question très simple en langage local. Le système opioïde, on a tendance à le voir surtout au niveau du système nerveux central. Est-ce qu'il n'est pas en fait un système réparti dans tout le corps et que le système opioïde est un besoin de majorité de nos cellules?
Marco?
So I don't know if I will answer correctly, and so forgive me for that. So for example, in the case of vaccine, the vaccines are selected for actually not binding any endogenous opioids. And so vaccination against, let's say, oxycodone will affect oxycodone, hydromorphone, and hydrocodone, but wouldn't affect any endogenous opioid system. So those will be preserved. So the baseline wouldn't be affected. I don't know if that was your question.
Thank you. Gavin, you can make your... Une question à Gavin Bart de ma part. Est-ce que tu as le sentiment qu'il y a quand même une... Yes? It's okay? Est-ce que tu as le sentiment que la crise des opiacés aux États-Unis est en train de se poursuivre, de se calmer, de décroître? Quelle est la tendance actuelle?
Well, we monitor very carefully, and hopefully we're reaching some slight plateau in deaths.
Okay.
What is clear is there is more adulteration of additional substances, and that multiple substance use, particularly combination with psychostimulants, is the increasing trend.
Okay. C'est une bonne nouvelle. Une dernière, et puis on va passer à la pause. Est-ce que quelqu'un a le micro? Non?
Thank you, Honora Englander from Portland, Oregon. Thanks for the great presentations. My question is around vaccine hesitancy, particularly in the US, where among people who use opioids, there's such intense hesitancy. You know, we see this with low uptake of medication for opioid use disorder, but I'm just curious how you or colleagues are addressing that in the vaccine development process.
Thank you.
Well, that's definitely a very valid question, and we've been asked about that, especially like in the United States and acceptance of the mRNA vaccine. One option that we are developing is also monoclonal antibodies. So just like we've seen with mRNA vaccines and COVID vaccines, those that are hesitant to actually get vaccinated seem to be open to other interventions such as monoclonal. So I don't know exactly if that's the full answer, you know, but at least we have like few options beyond vaccines.
Thank you. The last word for you, Ivan. And yes? To conclude this session. Yes.
Thank you so much. Well, you see all the many areas in development of treatments for substance use disorders, so vaccines and also small molecules. I just want to make a comment about the importance of the nasal nalmefene in the context of Gavin's presentation. Given the pharmacology of nalmefene, that's a new option that was recently approved by the FDA and started marketing like 2 weeks ago. So that I want to leave the message that that's a new option for treatment of fentanyl overdose with nasal nalmefene.
OK. Thank you, Ivan. Bon, merci beaucoup. Thank you very much. On a eu une chance inouïe d'avoir ces gens de grande qualité qui nous ont ramené des choses de grande qualité et qui ont répondu à notre questionnement. Thank you very much. Comme vous l'avez vu, on a pris du temps sur la pause, mais la bonne nouvelle, c'est qu'on a pu entendre des gens de qualité. La mauvaise, c'est que vous allez revenir à l'heure, à 11h pile poil. Donc, pour les copains qu'on n'a pas vus depuis 2 ans, on a le déjeuner. À tout à l'heure et à 11h.